Hey everybody, today we're going to learn about one type of biotechnology known as DNA fingerprinting. By the end of today's notes, you should be able to read and interpret a DNA fingerprint, which is what you see right here in the middle of the screen. So let's get started. So every single human being has their own individual unique set of DNA sequences, A's, T's, C's, and G's. And because everybody has their own unique DNA sequence, we can, as scientists, take that sequence and turn it into a DNA fingerprint, which, as you can see on the right, is a picture that depicts banding patterns that's made up of fragments of DNA. So here we have 5 different DNA fingerprints, and each of these little lines in this fingerprint represents a certain segment length of DNA. And it's that banding pattern that produces a fingerprint. We're not talking the fingerprint on your actual finger itself. So let's learn about how we make a DNA fingerprint. Well, we use a technology called gel electrophoresis, and you can see that machine there on the right. The device down here has a gel inside of it. There's some liquid, and we have a box that it hooks up to. This machine is really good at separating DNA based off of size, and by size I mean the number of DNA nitrogen bases in that segment, and also it separates it by charge. DNA has overall a slightly negative charge to it, and so this device in the background here, that's actually it's an electric current that we send through that solution, and the electricity produces that current and separates the DNA, because the DNA is going to be attracted to the slightly positive end of that electrical charge. Now, how do we get to the point where we can set up that machine and actually get the DNA fingerprint? Well, here's the process. First thing we're going to do is take a known sample of DNA, and we're going to expose it to a restriction enzyme. Restriction enzyme's function is to cut or cleave DNA into differently sized pieces. And it can do that because every restriction enzyme has a specific DNA sequence that it recognizes, like a certain ATCG sequence. And every time that restriction enzyme comes upon a DNA segment that has that particular sequence, it's going to cut in between the specific nitrogen bases. So we expose our DNA to that restriction enzyme, like scissors. Then we digest our DNA. And after it's digested and all chopped up, we then take a pipette and we suck up the DNA and we load it into a well of the gel. So that gel has these little holes in it, and those are the wells. And we're going to insert that gel with those DNA fragments. Then we're going to stick that gel inside the device, and we're going to turn on the electricity, and the electricity again is going to form that current, and that current is going to move the DNA fragments through the gel. Now, if you are a longer DNA fragment, you're going to run slower because you're heavier and the spaces in the gel are a little bit more difficult for you to fit through. It's like you leaving a crowded mall, and there's like 10 of you and your friends, and you're trying to get out of the mall when everybody's trying to come in. It's going to be a little bit difficult versus the smaller or the shorter fragments, the ones that only have a couple dozen nitrogen bases on them. They're going to run faster, which means they're going to move farther down the gel in the same amount of time. So then after we've exposed the DNA to this electricity for a certain period of time, we get that banding pattern formed. Now, individuals have different number of restriction sites. That's the sequence that the restriction enzyme recognizes. The number that I have versus the number that you have are going to differ because our DNA is different. So the number of times that the restriction enzyme chops up my DNA will differ from yours. And even if we had the same number of times that the restriction enzyme cut our DNA, the segments of DNA themselves would not be the same size. I might have more fragments that are longer than yours, or vice versa. And so because of that, our variation in DNA sequence and our unique codes allow us to have a very special and individualized banding pattern that can be used to compare my DNA with somebody else. Now there are three applications in which we use DNA fingerprinting. And the most common, and probably one that you might have already had some background information on, is crime scene identification. So if you are a forensic scientist and you go to a crime scene and you notice that somebody left their DNA at the crime scene, you can obtain that DNA, and then whatever suspects that the police officers came up with, you can go ahead and get samples of their DNA and then compare the sample from the scene to the suspects that you have. And the way that you identify which person did the crime is by comparing the banding patterns. If you look at the crime scene, this is the DNA that I extracted, I know that the person who committed the crime is going to have every single one of these bands match theirs. And we can see that suspect 1 is missing this top band here, this longest band, but we find it in suspect 2 and we miss it in suspect 3. Even though suspect 3 has a faintly light band here, it's not the same consistency as the one at the crime scene, and I can't change my DNA. So that means that suspect 2 was the one that committed the crime. Okay, the next application is going to be paternity. So say you are a woman and you claim that the father of your child is some billionaire, some millionaire, somebody with lots of money. Okay, well, the way to figure out if the child is actually the father's, because the mother's going to know it's her child, she's the one that gave birth, but to figure out who the father is, you're going to have to take the baby's DNA and you're going to have to compare it with the mother and the father, because all of the DNA that makes up that baby, half of it came from mom and half of it came from dad. So if this is our family, here's the mom, here's the dad, and we can go across and we can see daughter 1 has this band from the mother, this band from the father, this one from the father, and this one from the mother. So yes, in fact, she is their child biologically. Now if we go to daughter 2, this band is slightly above the yellow one. And of course we have the blue band that matches up with the mom, and so do those 2 blue ones, but this red one here did not match up with either mom and dad. So this daughter is biologically the mom's, yes, but not the father's. And we can look at son 2, okay? His matches up with the dad, his matches up with the mom, and so do the other ones. So he is in fact both of their child. Now son number 2, he's got light green and dark green bands. Neither are blue or yellow, and none of them line up exactly with the mom or the dad. So this is not their biological child. So son number 2 could have been adopted, right? He could have been a relative's kid that they, are fostering. Something else could have happened, like the baby was switched at birth, but he is, for whatever reason, not biologically the mom's and the dad's. Okay, so that's how we do paternity. The last is cataloging endangered species. So when a species is classified as endangered, meaning that their population is exceptionally low, scientists are going to go ahead and make them protected. So what we do when we make them protected is we go out and we take samples of their DNA and we store that genetic information in these DNA banks in order for us to go back later and test and analyze food, medicine, and other products to see if in fact people are protecting the species or illegally hunting and using them. So if in this case our endangered species was a bear, okay, and we have 3 bear samples and we have a food sample, we can go ahead and again compare the banding patterns. This white band right here, this is actually the well, so that's not actually a DNA band, but then all these gray ones are. So we can go ahead and again just match up the matching bands, and we see that the food sample actually matches up with bear 2. So was that food sample protecting that species? No. It could have been illegal hunting. It could have been the person didn't know the species was endangered, or it could have been they knew but they didn't care and they used it anyway. So that leads us to check yourself. I want you to take a minute and answer these 4 questions. What is a DNA fingerprint? What technology is used to make a DNA fingerprint? What type of enzyme is used to cleave the DNA? And 3 uses of DNA fingerprinting. So let's go through the first question. What is a DNA fingerprint? It is a unique banding pattern produced by separating DNA fragments. What technology do we use to make a DNA fingerprint? That's called gel electrophoresis. What type of enzyme is used to cleave the DNA? That's a restriction enzyme. And 3 uses of DNA fingerprinting. We have crime scene identification, cataloging endangered species, and my favorite, paternity testing. If you look at the bottom of your note sheet, you have this DNA fingerprint. From this, we need to determine the shortest pieces of DNA. If you recall from earlier, shortest pieces of DNA are going to be able to travel the fastest because they're not as heavy. The top of the gel is going to be here near the names, and the bottom of the gel is going to be farthest away. So if we were running this gel for 30 minutes, the shortest piece would be able to run the farthest down the gel. So the shortest piece would be this one down here at the bottom. That means that the longest pieces of DNA are going to have to travel the shortest distance in the 30-minute time period. So if we started here at the top near the names, then the longest pieces are going to be found closest to those names, closest to those wells, and they're up there. Okay. Now, the last part that you have to do for this is determine who committed the crime. Now, we can see that we have blood on the knife, 1 victim, and 2 suspects. You will have to go ahead and compare the victim's DNA with both of the suspects' DNA. And we need to then compare it with the blood on the knife. Because if you see, no single person here, the victim or the suspect's DNA fingerprint matches exactly the blood on the knife, which means that that is a contaminated sample of blood. So it contains victim's blood because they were stabbed, and then perhaps the suspect also cut their hand in the process. So if we can say that this first band came from the victim, this 4th band came from the victim, and so did the 5th band, that means that we are looking for really these 2 bands. So if we compare, the 2nd band is missing from suspect A, but present in suspect B, which is a good sign. The 3rd band is present in both suspect A and B. Okay. And then we can see that suspect B also has this DNA band all the way down here that also matches up with the victims, which is perfectly fine. So based off of that information, and we can eliminate suspect A because they have this piece that we've already highlighted, which is missing from the blood on the knife. And again, you can't change your DNA or your fingerprint. So that means that person B committed the crime.